Hello, learners. This is Navnath Pawar, Assistant Professor, Department of Zoology, Thakur College of Commerce and Science, Kandivali. So today's topic is mutation. In today's lecture, we are going to deal with the mutation. So I hope you all are aware from these kinds of words: mutation, change, or anything else. Just like you can say something is there, some changes are there, right? So you can say ki we can afford change in your mutations are nothing but a sudden change in genome. So first of all, uh, let's discuss on what is meant by genome. So what is meant by genome? Genomes are nothing but haploid set of chromosome. So there are so many reasons for the mutation, for the change in genome, for the change in nucleotides, DNAs, RNAs, or protein sequence. So out of them, some parts in today's lecture we are going to deal with them. So this is just introductory part of what is meant by mutation. So simply you can say here mutations are nothing but sudden change in genome, and genome is nothing but haploid set of chromosome, or haploid set of chromosome is called as genome. And in genome there will be a change due to various reasons, and that is called mutations. So first of all, what are mutations? So now you can see here change in the nucleotide sequence of DNA. So first of all, what is meant by nucleotide? Then you can learn about the change in nucleotide sequence of DNA, right? So nucleotides are, you can say, nitrogenous base plus sugar plus phosphate group present in the DNA. They are called as nucleotides. And what is meant by nucleoside? So nucleotide minus phosphate group, now you can say it's nucleoside. So change in the nucleotide sequence of DNA is called as mutations. Now here you can say may occur in somatic cell. Means in our body there are two types of cells. Number one, somatic cell, and number two, germ cell. So let's discuss mutations happens in the somatic cell, and let's discuss mutations are happens in the germ cell. So what will be the result after mutation in somatic cell, or what will happen after in germ cell after mutation in germ cell? So let's discuss first of all in about somatic cell. So somatic cells means what? Somatic cells are there for growth and development of individual. It's called somatic cell. And what is meant by germ cell? So germ cells are reproductive cells. Means further they are converted into the gametes. So let's discuss if mutations occurs in the somatic cell, then what will happen? If mutations are occurs in somatic cell, then only that individual will suffering from that kind of effect. It's called somatic cell mutation. And if mutations occurs in the germ cell, then what will happen? If mutations occurs in the germ cell, then F1 generation will suffering from that kind of effect. So that's the basic difference between somatic cell mutations and germ cell mutations. Now, here you can say gametes, or you can say germ cells. If mutations occurs in the germ cell, they will be affect to the next generation, and if it is in somatic cell, they will affect that particular individuals. That is the basic difference between somatic cell mutations and gametic cell mutations. Now here is the topic: are mutation helpful for harmful? Right, most of time we know the mutations are harmful for all of us, right? But sometime mutations are helpful for us. Why helpful for us? When that mutations occurs in the animals or in, you can say that, individuals, then that will be harmful for us. But if it is occurs in the plant cell, then it will be helpful for all of us. So let's discuss on it. Mutations happens regularly. Why? Because as we know, we are continuously contact with the X-rays, gamma rays, sun rays, ultraviolet rays, various types of pathogens, various types of chemicals, various types of mutagens. So what is meant by mutagens? Now here we can say mutagens are the agent to cause the mutation, right? Mutagens, what is meant by mutagen? Mutagens are the agent to cause the mutation. So it may be different kinds of mutagens are there. It may be in form of rays, in form of chemicals, in form of viruses, in form of DNA elements which are present in the animal and plant cell as well. And that kind of mutations are repaired by many enzymes which are present inside the individuals. What then? Some type of skin cancer and leukemia result from somatic mutations. As I told earlier, if mutations occurs in the somatic cell, then what will happen? That specific individual will suffering from that kind of effect, right? For example, skin cancer. For example, leukemia. So leukemia is basically related to the WBC, white blood cell. So if we are suffering from somatic cell mutation, then there will be a chances that particular individual will suffering from skin cancer and leukemia. Some mutations may improve an organism survival. For example, the mutations which is beneficial for us. So let's assume that that particular individual already suffering from mutations, and due to another mutation on same place, that existing mutation will be recovered, and now you can say that kind of mutation is beneficial for us. Here we can say what is a chromosome. Quick review, because as I told you, what is meant by genome? Genomes are the haploid set of chromosome, and what is meant by mutation? Sudden change in genome, right? So for better understanding, we have to know about the chromosome. So here you can see chromosomes is a DNA molecule that is tightly coiled around proteins called histones, which support structure and to form thread-like structure. So here you can see in the diagram a DNA double helix structure, chromosome, P arm. Short arm is called as P arm, long arm is called as Q arm, and here are some proteins which is there for binding, and that is nothing but the histone protein. Then let's move towards the main topic, types of mutation. So what are the types of mutation? So first of all, let's discuss on chromosome mutation. Means what? Mutations occurs at chromosomal level. So you can see here changing the structure of chromosome or the loss or gain of part of a chromosome, that will also lead to the chromosomal mutations. For example, there are five types of mutations are there. Number one, deletion type of mutation, inversion type of mutation, translocation type of mutation, non-disjunction type of mutation, and duplication type of mutation. So these are the five types of chromosome mutations. So let's discuss one by one in detail. So first, deletion. So due to breakage or delete of some part of chromosome, or you can see here a piece of chromosome is lost. So now you can see here in the picture, here is the original chromosome, and due to deletion some part get deleted, and now this resulting structure is here. So due to deletion of a chromosome, the change will be there in the genome, and that kind of mutations are called as deletion type of mutations. Means some part is going to delete from the chromosome, and due to deletion type of chromosome, result will be different. It may be change in characters, change in protein sequence, change in genome sequence, right? But due to some part get delayed, that kind of effect or that kind of mutations are called as deletion type of mutations. Now second, inversion. So what is meant by inversion? So see here, chromosome segment breaks off, segment flip around backward, and segment reattach. So you can see here in the picture, breaks off chromosome, and after breakage they will get reverse, or you can say inverse. Now here you can see the green part of chromosome is at lower side and red part of chromosome at upper side, but due to inversion type of mutation, the result is, you can see here, green part is on the upper side and red part in lower side of chromosome. So why this is happens? Because of inversion type of mutations. So these kinds of mutations also leads to the problems, or you can say effects, in the morphological character, in the genetical character, or in the sequence of genome, sequence of nucleotides, sequence of DNA. So these kinds of things are there. So that's why change will be there. Now duplications. On the basis of words, you can say here duplications means duplicate copy. So formation of exact copy is called as duplicate, right? Occurs when a gene sequence repeated. Means, for example, let's assume that ATGC is the original sequence of DNA. Okay, and after duplication, ATGC duplicate, formation of exact copy, that is called duplication. Okay, so this is just about duplication. Now translocation. Translocations means what? Now you can see here, involves two chromosomes that are not homologous. What is meant by, now here new terminologies are here, homologous. So what is meant by homologous? So homologous means what? Homo means same. So homologous chromosomes are nothing but same chromosome. And now for translocation, some part of one chromosome is transferred to another chromosome. That is called translocation. Here you can see in the picture, before translocation and after translocation. So here is the entire part of chromatid or chromosomes, and here is the second chromosome which is not homologous. And after translocation, you can see here there is a change in their structure, change in their morphology. So what is meant by translocation? Translocations means simply you can say transfer of one part of chromosome into second chromosome. That is called translocation type of mutation. Now non-disjunction means what? Non-disjunction is the main reason behind mutations, right? So what is meant by non-disjunction? Now you can see here, failure of chromosome to separate during meiosis. So I hope you all are aware from the terminologies meiosis and mitosis. So what is meant by mitosis and what is meant by meiosis? What is meant by mitosis? So mitosis is the cell divisions which occurs in the somatic cell. And what is meant by meiosis? Meiosis is a kind of cell division which occurs in germ cell. So meiosis is there for germ cell division and mitosis is there for somatic cell division, right? But at the time of meiosis, if there is a failure of chromosome at the time of separation, now things will be different. right? So that's called non-disjunction. For example, non-disjunction causes the chromosomal disorders. For example, Down syndrome. So what is meant by Down syndrome? So Down syndrome is the trisomy of 21st chromosome. Means in normal individual, there is a pair of chromosome, right? One from mother, one from father. One is paternal chromosome, one is maternal chromosome. But at the time of non-disjunction, at 21st chromosome is repeated. Means instead of 2, there will be a 3 chromosome, and that is called Down syndrome. For example, next example, Turner syndrome. So Turner syndrome, single X chromosome. So as you know, how many types of chromosomes are there? Two types of chromosomes are there. Number one, autosome, and number two, allosome. Right? So allosomes are also called as sex chromosome. Right? XX which are present in the female and XY which are present in the male. Turner syndromes are occurs in the female. Means instead of XX chromosome, after non-disjunction, instead of XX, there will be a X0. Means what? At the time of gamete separation, non-disjunction will be there. And that is why instead of XX, there will be a X0 chromosomes, which are mostly happens in the female. That is called Turner syndrome. And for male, here is you can say second example, Klinefelter syndrome. Right? So in case of Klinefelter syndrome, double XY. Yes, along with XY, extra X is there. And Klinefelter syndrome is mostly observed in the male. So Turner syndrome is observed in the female, and Klinefelter syndromes are observed in the male. Okay. So let's see normal male. So this is the karyograph. After karyograph, in case of normal male, the picture will be like this. Normal, all chromosomes are normal. And in case of female, you can say, right? So this is the normal karyograph for female. Now, yes, now you can see here, Down syndrome. What will happen after Down syndrome? So here I mentioned Down syndrome is also known as trisomy of 21. Why they are called as trisomy of 21? Because 3 chromosomes are there, right? And it's a genetic disorder caused by the presence of all or part of third copy of chromosome 21. And due to these kinds of syndrome, it is associated with the physical growth delay, characteristics of facial features, and other disabilities are there, right? Then, yeah, you can see here, male trisomy 21 Down syndrome. So where is the 21 chromosome? Here, you can see here. 3 copies of chromosome, right? Here, this one. 3 copies of chromosome. That's why they are called as trisomy of 21. So, after karyograph, you can see there is a trisomy of triple chromosome at number 21. That's why they are called as trisomy 21, which is also called as Down syndrome. Now, Turner syndrome, as I told you, Turner syndrome mostly observed in the female, or you can say only female, right? A condition that affects only female results when one of the X chromosome is missing, right? As I told you earlier, what is meant by Turner syndrome? In case of Turner syndrome, 22 pairs of chromosome plus, instead of double X, X0 is there. which are only observed in the female. And what will be the effect after deletion of that chromosome or after that X0 type of syndrome? Now you can see here variety of medical and developmental problems, including short height, failure of the ovaries to develop, and heart related problems. This is due to the X0 or due to the Turner syndrome. Yeah, you can see here normal as compared to the normal karyograph. All things are normal, but in case of sex chromosome or allosome, you can see here only one X chromosome is here, and one X chromosome get separate at the time of meiosis, right? So that's why instead of XX, you can see here only single chromosome is there, which is X. And on the basis of these karyograph, you can say that particular individual is suffering from Turner syndrome. Now Klinefelter syndrome, as I told you, which are observed in only in males, means in normal males XY chromosome is there, right? But after Klinefelter syndrome, instead of XY, there will be a XXY, right? Now you can see here what will be the effect after Klinefelter syndrome. Now physical traits, weaker muscles, greater height, poor coordinations, less body hairs, and sterility. Sterility means they are not able to form new young ones, right? Sterility. So these are the effects after suffering from Klinefelter syndrome, which are observed in only male. Okay, now here you can see karyograph for male, right? Rest of the things are same. 22 autosomes are same, right? But here you can say here you can see XXY. So double X plus Y. This indicates that individual is suffering from Klinefelter syndrome. So after suffering of Klinefelter syndrome, how many chromosomes are there? Total number of chromosomes are there in normal male or in normal female? 46 chromosomes are there, right? But when they are suffering from Turner syndrome, the number of chromosome is 45. Why? In case of Turner syndrome, one chromosome get delete, right? Or separate. So total number is 45. And in case of Klinefelter syndrome, here is a one extra X chromosome. So instead of 46, here you can say 47 chromosomes in case of Klinefelter syndrome. Yes. Now here is the animation, chromosome mutation animation. Now you can see one by one deletion, inversion. Here is the translocation. Here is the duplication, right? And here is the original sequence. So this animations indicates that how chromosome get mutant due to duplication, due to inversion, due to deletion, due to translocation. Yes, here you can say the first is the original chromosome, right? After duplication, you can see here AB, AB, CC as it is, DD. Yes, here is the extra D, means duplication of existing D is there, right? This is called duplication. Now next deletion, you can see here D get delete, right? So deletion. Inversion means change their position, right? So you can see here F in normal chromosome EF and in inversion chromosome FE, right? And here, yes, you can see here also ACB, D, E, F, G, and this is the normal sequence. So if you compare with the original chromosome, we can see there duplication type of things are there, deletion types of things are there, inversion types of things are there. So single picture indicates all the things: duplication, deletion, inversion. Now gene mutation. So change in nucleotide sequence of a gene. So first of all, tell me what is mean by gene. Most of students says that genes means what? Piece of DNA, piece of chromosome. And actually that definition was wrong. Why? Because not all parts of chromosomes are called as gene, not at all, but only functional part of DNA, functional part of chromosomes are called as gene. Right? So change in the nucleotide sequence of a gene. So I hope you all are aware from the nucleotide, right? So nucleotide are the, yes, nitrogenous base plus sugars plus phosphate. Combinedly they are called as nucleotides. Right? May only know a single nucleotide means due to some kind of defect that single nucleotide will get changed. Right? Or maybe more than one nucleotide also. Right? Yeah. So you can see here due to copying errors means at the time of replication, due to contact with the chemicals, due to contact with the viruses, that particular individual will suffering from the different kinds of mutations. Okay. Now see here different types of mutations, different types of gene mutations. So first part was types of chromosomal mutations, and here you can say types of gene mutation. So total types of gene mutations are 8. Point mutation, sense mutation, missense mutation, fourth one nonsense mutation, fifth one insertion, sixth one deletion, seventh one frame shift, transition and transversion. So these are the types of gene mutation. Okay, so let's discuss one by one. First, what is meant by point mutation? So point mutations means what? Change in single nucleotide. Doesn't matter whether it's due to deletion or due to inversion or due to insertion or due to substitution. Doesn't matter. If change in single nucleotide, you can blindly say there are top point mutations are there. Right? So you can see here in the screen includes the deletion, insertion or a substitution of one nucleotide in a gene. They are called as point mutation. Keep it in mind once again. What is meant by point mutation? Only single nucleotide getting changed. That is called point mutation. Here is the one example for point mutation. Sickle cell anemia, or you can say sickle cell disease. Basically that sickle cell anemia is caused due to the hypoxic condition. But what is meant by hypoxic? Low oxygen level. But due to low oxygen level, the gene which is there for thalassemia, that will get changed, and that is called sickle cell disease. Means you can say it occurs in the hemoglobin gene. Means only single nucleotide get changed in the hemoglobin gene. After that, that particular individual will suffering from sickle cell anemia, sickle cell disease, and this is the example for point mutation. Sense mutation. So what does mean by sense? Right? So you can see here, even after change in nucleotide and codon, the same amino acid occurs at same position. Means sense mutation. Example, here you can see all the sequence is same, CCA, that A is converted into the C after mutations. Means that CCA codon is code for proline amino acid. What I am saying? That CCA is code for proline amino acid, and after getting change, instead of A, there is a C. So triple C is also there for proline amino acid. So means after change in the sequence of nucleotides, there is a no change in the amino acids, right? That kind of mutations are called as same sense mutations. Yeah, mutations are there, but there is no effect on proteins or amino acids. Missense mutation. What is meant by missense? So same things are here, but instead of same amino acid, you can say hydrophobic amino acid is replaced by hydrophobic amino acid, and hydrophilic amino acid is replaced by hydrophilic amino acid. It's called missense mutation. So what is meant by hydrophobic amino acid and what is meant by hydrophilic amino acid? Hydrophobic amino acids are nothing but water hitting amino acid, and hydrophilic means water living amino acids. So here are the some examples: glycine, alanine, valine, leucine, isoleucine, proline, phenylalanine, methionine, and tryptophan. So if hydrophobic amino acid is replaced by hydrophobic amino acid after mutation, now you can say missense mutation. Hydrophilic amino acid is replaced by hydrophilic amino acid is also called as missense mutation. Now, yes, nonsense mutation. So for better understanding about the nonsense mutation, we have to know about stop codons or nonsense codon. Why they are called as stop codon? Why they are called as nonsense codon? Because after having that codons, for example UAA, ochre, the name of that codon is ochre, UAG, amber codon, UGA, opal codon. So these are the three stop codons and they are there for terminate the protein synthesis. What I am saying? These three codons are there to terminate the protein synthesis. And after termination, immature protein will be formed, non-functional protein will be formed, and that's why they are called as nonsense mutation. For example, here we can say AAA, GGG, UAU. Okay, these are the normal sequence. And after mutation, that U is replaced by U is nothing but uracil. Uracil is replaced by guanine. So this is the functional form of protein. But after change, there is a formation of nonsense codon or stop codon. And that stop codon leads to a, yes, to stop the synthesis of that protein. So what will happen after that? Immature protein will be formed. And if it is immature, then it is non-functional. Right? That's why they are called as nonsense mutation. Right? Yes. Insertion mutations. Insertion mutations means simply addition of nucleotides. That is called insertion type of mutation. So here you can see in original sequence and in after insertion of sequence. So extra C is there. Extra C stands for cytosine. Right? So extra, if it is extra, if it is, someone is there to get add in the normal sequence, now you can say insertion type of mutation. Okay? Yes. Deletion. Yes. Same just like insertion, but instead of addition, now you can say here deletions. some part get deleted from the original sequence. What I am saying? Some part get deleted from the original sequence is called as deletion type of mutation. Now, frame shift mutations. Now, first of all, what is mean by frame? What is mean by shift? And what is mean by mutation? So, what is mean by frame shift? So, what is mean by frame? The sequence which is started from start codon and ends with stop codon. That is called frame. ORF, open reading frame. So, what is mean by open reading frame? The sequence which is start with started codon and end with stop codon. They are called as ORF, open reading frame. Okay, but that frame will shift after insertion of the nucleotides or deletion of the nucleotides. Let's see how. Yes, let's assume that the fat cat at the wee rat is the original sequence of protein. Let's assume that. Okay, and in original sequence one extra A get add. What will happen? Instead of CAT, here we can say CAA is here. AT instead of ATE, TET is here. Instead of THE, HEW is here. Right. So what will be the result after getting addition or deletion of nucleotides? That entire frame will shift. And that's why they are called as frame shift mutation. Due to addition of nucleotides or deletion of the nucleotides. Doesn't matter whether it is single nucleotide get add or triple nucleotide get add or 4 nucleotide get add, right? But if triple nucleotide get add, then frame also shift. Okay, so these are the examples of frame shift mutation. Now here, yes, exact example for frame shift mutation. In normal sequence, methionine, glutamine, alanine and glycine amino acids are there. But after getting addition of extra Z, methionine, lysine, histidine amino acids is there. Right? So this leads to a change in the frame. Right? And that's why they are called as frame shift mutation. Next, yes, substitution, insertion, deletion, inversion. So this is a gene mutation animation. Okay? Yes. Yes, most important part. Transition mutation and transversion mutation. So for better understanding about the transition mutation and transversion mutation, we have to know about types of nucleotides. What I am saying? Types of nucleotides. So can you tell me how many types are there? So basically, nucleotides are categorized into two types. Number one, purine, and number two, pyrimidine. So purine nucleotides and pyrimidine nucleotides. Now how many nucleotides are there? 5 nucleotides are there. What are those? First adenine, second thymine, cytosine, guanine and uracil. Right? These are the 5 nucleotides and they are categorized into how many types? Two types, purine and pyrimidine. So out of 5, 2 nucleotides are belongs to the purine and 3 nucleotides are belongs to the pyrimidine. Right? So what are purines? Adenine and guanine are the purine molecules. And what are the pyrimidines? Cytosine, uracil and thymine. These 3 nucleotides are the pyrimidine type of nucleotides. Now let's come to the point, transition type of mutations. right? So what is meant by transition mutation? So here you can say purine nucleotides is replaced by purine nucleotide, or pyrimidine nucleotide is replaced by pyrimidine nucleotide, right? Here you can see uracil is replaced by cytosine, both are pyrimidine. Adenine is replaced by guanine, both are purine. So that's why they are called as transition type of mutation. Purine is replaced by another purine, pyrimidine is replaced by another pyrimidine. And here is the example. Okay, so these are nothing but the transition type of mutation. Let's move towards the next mutation, transversion type of mutation. Yes, just like a transition type of mutation, but instead of purine is replaced by purine, you can say here purine is replaced by pyrimidine nucleotides, or pyrimidine nucleotides are replaced by purine nucleotides, right? How? This is the example. Adenine is replaced by cytosine. Adenine is the purine, cytosine is the pyrimidine nucleotide. Here is the cytosine is the pyrimidine nucleotide, guanine is the purine nucleotide. So purine is replaced by pyrimidine, or pyrimidine is replaced by purine. So these kinds of mutations are called as transition type of mutations. Yes, thank you. Now this is all about the chromosomal mutation and gene mutation. But in next lecture, we will discuss on how to repair that mutation, by artificially, by naturally. Thank you all.